All right, let's jump into a world that's invisible to the naked eye, but has a huge impact on scientific research and medicine mammalian cell cultures.
Ooh, interesting.
These tiny cells hold incredible potential, but they're also incredibly vulnerable.
Yeah,
It's like trying to raise a delicate seedling in a hurricane, except the hurricane is microscopic and filled with bacteria and fungi.
that's a great analogy.
So we're talking about a constant battle against invisible enemies.
Exactly.
What are some of the biggest threats in this microscopic world?
Well, bacteria are a major concern.
Okay.
They're everywhere, and they multiply so rapidly, competing with your cells for nutrients.
Yeah, so they're taking all the food.
Right. It's like having a tiny, uninvited guest at a dinner party who proceeds to eat everyone else's food.
Oh, no.
And then there's fungi, which can create these sprawling, unsightly growths. Imagine a jungle overtaking your carefully manicured garden.
That sounds awful.
Yeah, and then we have the stealthy mycoplasma, a type of bacteria that can be incredibly difficult to detect.
Oh, really?
They're like microscopic ninjas that can sabotage your experiments without you even realizing it.
So it's not just about aesthetics, it's about ensuring the integrity of the research itself.
Exactly.
It sounds like maintaining sterility is absolutely crucial.
It is.
What's the first line of defense in this microscopic battle?
The cornerstone of any sterile work environment is the laminar flow hood.
Okay.
It's essentially a fortress against contamination.
Okay.
These hoods use a HEPA filter, which stands for high-efficiency particulate air.
Mm-hmm.
Think of it like a super fine mesh that can trap particles hundreds of times smaller than a human hair.
Wow.
This creates a constant stream of clean filtered air flowing over your work area, pushing away those pesky microbes trying to invade.
So it's like a shield protecting those vulnerable cells from an onslaught of microscopic invaders.
Precisely.
But I imagine not all laminar flow hoods are created equal, right?
That's right.
So tell me about it.
The most common type used in cell culture is the Class II hood, which provides a dual layer of protection for both the person working and the cells themselves.
Interesting.
It's designed to direct airflow inward and downward, creating a sort of invisible curtain that keeps contaminants at bay.
It sounds like using a laminar flow hood correctly is a key skill in this field.
Absolutely.
What are some of the essential things to keep in mind when working under the hood?
Well, before you even start working, cleanliness is paramount. Think of it like prepping a surgical field. You need to meticulously wipe down the entire hood with 70% ethanol or a suitable disinfectant, ensuring every surface is spotless.
Wow, that's a lot of cleaning.
Yeah, and don't forget to turn on the hood at least 10-15 minutes beforehand to allow that sterile airflow to stabilize.
So it's like creating a clean slate and letting the hood do its magic before you even introduce those delicate cells?
Precisely.
Yeah.
And once you're ready to work, it's all about controlled movements and minimizing any disruptions to that precious airflow. Avoid sudden gestures or reaching across sterile surfaces.
Gotcha.
Think of it like conducting an orchestra. Every movement should be deliberate and purposeful.
Okay, so we've got our protective barrier in place, the laminar flow hood.
Right.
But what about the tools and equipment we use inside?
Yes.
How do we ensure those are free from contaminants as well?
That's where sterilization methods come into play.
Okay,
And luckily, we have a whole arsenal at our disposal.
great.
The workhorse of sterilization is the autoclave,
Okay,
which uses high-pressure steam to obliterate any lingering microbes.
so it's really getting in there.
Yeah, imagine a pressure cooker on overdrive.
Oh.
It subjects materials to temperatures of 121 degrees Celsius for 15-30 minutes,
Oh, wow.
essentially cooking those microbes into oblivion.
That's intense. But wouldn't that damage delicate equipment?
It's certainly not suitable for everything.
Okay.
You always have to check if an item is labeled as autoclave safe. Glassware, metal tools, and certain types of plastics can handle the heat, but other materials might melt or warp.
I see.
For those more sensitive items, we can turn to chemical sterilization.
So there are different sterilization methods for different materials.
Exactly.
What are some of the options when an autoclave is not suitable?
Chemical sterilization uses gases like ethylene oxide or formaldehyde to effectively eliminate microbes.
Okay.
It's a bit more specialized and requires specific safety protocols,
Mm-hmm.
but it's essential for sterilizing heat-sensitive items like delicate plastics or electronic components.
That makes it. It's like having different weapons in your arsenal to target specific enemies.
Right.
Are there any other sterilization methods commonly used in cell culture?
We also use UV sterilization, which is fantastic for surfaces.
Oh, yeah, I've heard of that.
It relies on ultraviolet light to damage the DNA of microbes, effectively killing them.
So the light just zaps them?
Pretty much.
That's cool.
You often see UV lamps used inside laminar flow hoods or biosafety cabinets to maintain a sterile environment. But keep in mind that UV light doesn't penetrate deeply, so it's best suited for surface sterilization.
Okay, so UV light is like a surface weapon, while autoclaving and chemical sterilization can target microbes hiding within objects.
That's a great way to think about it.
What about liquids and gases? How do we ensure those are sterile?
That's where filtration comes in.
Okay.
It's a gentler method, perfect for sterilizing liquids and gases that would be damaged by heat or chemicals.
Mm-hmm.
It's all about using a membrane with incredibly tiny pores, typically around under 0.2 micrometers, to physically trap any unwanted microbes.
So it's like a filter.
Yeah, imagine a microscopic sieve allowing only the purest substances to pass through.
I like that analogy.
Filtration is a crucial part of maintaining sterility.
Okay, so we've covered the laminar flow hood and various sterilization methods,
Right.
but I imagine maintaining a truly sterile environment extends beyond just these tools and techniques.
Oh, absolutely.
What else is involved in creating a culture of sterility, so to speak?
That's a great question.
Thanks.
And you're absolutely right.
Well, sterility is not just about having the right equipment. It's about cultivating a mindset, a constant awareness of potential sources of contamination, and a dedication to meticulous practices.
That makes sense.
It starts with basic hygiene,
Okay.
always wearing lab coats, gloves, and masks to prevent contamination from ourselves.
Good point.
Our hands, even our breath, can carry microbes that could jeopardize those delicate cells.
So it's a reminder that we ourselves are often the biggest potential source of contamination.
Absolutely.
What else contributes to maintaining a sterile environment in the lab?
Air quality is critical.
Okay.
We use air filtration systems to ensure the air circulating in the lab is clean. And for highly sensitive work, we might even use biological safety cabinets,
Oh, wow.
which offer an even higher level of protection than a standard laminar flow hood.
So these are like super hoods.
Yes, think of it as creating a clean room environment, minimizing any potential for airborne contaminant to settle on surfaces or infiltrate our cultures.
That's pretty intense.
And of course, general lab cleanliness is paramount, so keeping things tidy.
Exactly. A well-organized lab, free of clutter and debris, goes a long way in preventing contamination. Regular wipe-downs of surfaces with disinfectants are essential, as is careful handling and disposal of contaminated materials.
It sounds like maintaining a sterile environment is a multifaceted endeavor
It is.
involving a combination of specialized equipment, meticulous techniques, and a constant awareness of potential threats.
Yes, definitely.
But let's be realistic for a moment.
Sure.
Even with the best intentions and rigorous practices, contamination can still happen, right?
Unfortunately, yes. Despite our best efforts, those microscopic invaders can sometimes find a way to breach our defenses.
So what do we do?
That's why it's crucial to be able to identify contamination quickly and take swift action to contain and eliminate it.
Okay, so what are some of the telltale signs that a culture has been compromised?
Right.
What should researchers be on the lookout for?
It's all about observation.
Okay.
You need to be vigilant, inspecting your cultures regularly for any changes in appearance or growth patterns.
Mm-hmm.
If the liquid surrounding the cell suddenly turns cloudy, that's a red flag, often indicating bacterial contamination.
Oh, that's interesting.
Or if you see fuzzy growths,
Ah.
those are likely fungi setting up shop.
Oh, yeah.
One of the trickiest contaminants to detect is mycoplasma.
Okay.
These tiny bacteria can be incredibly stealthy, often not causing any noticeable changes in the culture's appearance, but they can subtly alter the behavior of your cells, leading to misleading results.
So it's like playing detective trying to spot subtle clues that might indicate a microbial invasion is underway.
That's a great way to put it.
What are the next steps if a researcher suspects their cultures have been contaminated?
Swift action is key.
Okay.
The first step is to isolate the contaminated cultures to prevent the spread to other cultures.
Right, so quarantine them.
Exactly. You need to dispose of them properly, usually by autoclaving them before putting them in biohazard waste.
That makes sense.
Then you need to thoroughly disinfect the entire lab, including all surfaces equipment and, of course, the laminar flow hood.
Wow, so a deep clean.
Yeah, it's like hitting the reset button, ensuring a clean slate for future experiments.
So even with all these precautions, it's almost inevitable that contamination will occur at some point.
It can happen, yeah.
It sounds like it's not just about preventing contamination, but also about having protocols in place for swift,
and effective remediation.
Precisely. Maintaining sterility is a constant process requiring diligence, attention to detail, and a commitment to upholding the highest standards of cleanliness in every aspect of the work.
Sounds like a lot of work.
It is a challenge, but it's a challenge worth conquering to ensure the integrity and reliability of scientific research.
This has been an incredibly insightful look into the world of aseptic techniques.
It's fascinating, isn't it?
We've covered a lot of ground, from the invisible threats to the tools and techniques used to combat them.
Absolutely.
It's clear that mastering these techniques is essential for anyone working with cell cultures, ensuring reliable results and contributing to the advancement of science.